Hello. So we'll continue our lectures and talk about how will medical biotechnology change our lives over the years ahead. One of the major advantage of medical biotechnology is the microarray or the gene chip. So how you can test one person's DNA for many different SNPs or the single nucleotide polymorphisms. You isolate the DNA from the patient. First you collect blood, and then you isolate DNA from the blood. Then apply this sample to a microarray which contain many DNA sequences. And then you compare the patterns of DNA binding between patient's DNA and the DNA on the microarray to reveal the patient SNPs pattern. What about pharmacogenomics, which is the customized medicine? Actually, it's you design and tailor a drug therapy and treatment strategies based on the genetic profile or the genetic background of a patient. So you need to do a microarray analysis, then design drugs against genes that are mutated for an individual patient. And that's why it's called customized medicine. What about metabolomics? Actually, it comes from the metabolites that our body has due to chemical reaction. So it is like a snapshot of the small molecules produced during cellular metabolism. And you can distinguish between disease process and physiological adaptation according to change in these small molecules compared by a normal person. So you can compare between this small molecule produced from a normal person and diseased person, and you can see how the metabolomics will change, and it will give a clue about the disease. What about nanotechnology? Actually, nanotechnology comes from the nanoparticles, which is a small, very minute particles in the range of nano that can deliver drugs to the cells. And you can see from the figure here, this is lipid nanoparticle containing the therapeutic drug, and you can inject it to a patient that have cancer, and it have high efficiency rate to reach to the cancer cells when it's coated by the drug. What about gene therapy technology? Actually, it aims to replace the defective gene with normal copy of a gene, but it still have barriers to overcome before this technology comes safe and effective. Okay, because what worries here that you don't do the process properly, and you will deliver the gene in another place. So the obstacles include how can normal genes can be delivered to all cells in your body? What are the long-term effects of introducing extra genes in humans? What must be done to ensure that the proper protein is made? So you can already insert the gene, but how can you be sure that the proper protein will be made from these genes? What about small interfering RNA, or simply siRNA? This is a technology is used to silence genes that are involved in disease progression. For stem cell technology, most stem cells are from embryos, which is called embryonic stem cells. But they are controversial since the process involves the death of an embryo. So in other words, if you want to take embryonic stem cells, you eventually will lead to the death of the embryo. For the other stem cells from adult stem cells, we call it adult stem cells. And either type of these stem cells can be coaxed to grow into cells of interest to replace damaged cells or failing organ, like any organ of your body. So the main aim of the scientist is to use these stem cells to make it differentiate into specific type of cell and then form a specific type of tissue like liver and pancreas. And they use these differentiated cells and tissues to retransplant back to the same individual so that they can avoid the problem of rejection of transplantation. And you can see here from this simple figure that the embryonic stem cells can be differentiated into many different types if you know the correct condition of differentiation. Actually, they don't know up till now most of the cell how they differentiate. And as I told you, the main aim is once these cells are differentiated into the specific intended type of cell, you can transplant it back to the individual to replace any damage or a defective tissue. So what about regenerative medicine? The regenerative medicine or genetically modified stem cells of patient, we try to genetically or the scientists aiming to use the genetically modified stem cells of patient to treat genetic disease conditions. In future, scientists will be able to isolate adult stem cells from a patient with a genetic disorder. Then they genetically manipulate these cells by gene therapy approaches, and then reinsert these cells back into the patient to help treat their genetic diseases. So let's continue for the biotechnology workforce. We need to differentiate between a biotech company and a pharmaceutical company. Actually, a pharmaceutical company is involved in drug development. They purify compounds to make a drug. They chemically synthesize drugs. The pharmaceutical company doesn't use living organism at all to grow or produce a product or a drug. While a biotech company is involved in drug development, must use a living organism or a product from a living organism. The biotech company also create products that are not drugs. The biotechnology industry provides very exciting opportunity for many types of scientists, from biologists, biochemists, to engineers, to ITs, to sales people. This industry of the biotechnology offers a great range of high-tech employment opportunities. So gaining research experience as an undergraduate is an excellent way to prepare yourself for a career in biotechnology. And you can see here from the figure, how in what branch in a biotechnology company is needed and what qualification is needed. So you can see we have 4 branches in a biotech company. You have the analysis section, you have the production section, and you have the QA/QC, quality assurance, quality control section, and you have the RD section. If you have a level of graduate of a high school graduate, so probably you will work in the production section. Most of the only having high school degree will work in the production section. You can see that if you are graduate, you have a graduate degree, that's mean like a master degree. So mostly you will go into the RD section, the RD, which means research and development. And the least will go for the production sector. For a bachelor degree, mostly will go for the R&D as well, together with the analysis section or a sector. So actually, from here we can see the minimum level of education required. And for R&D, actually, specifically, requires more training and greater amount of training over the other jobs. And you can see these table as well. The top five biotechnology companies are the top five pharmaceutical companies. You can see that these companies are different. And the revenues, what they gain, what they earn, what they got, what they from selling their products. Okay. Now we'll talk about the third topic of the biotechnology, which are genes and genome mutation and causes and consequences. So let's review the cell structure. Review, just make it fast. And then we'll talk about the molecule of life, which is actually the DNA. Then we'll talk about the chromosome structure, DNA replication in genomes. Then we'll talk about the RNA and protein synthesis. Then we'll talk about the mutation and causes and consequences. Of course, most of these data, this info, you might have taken in other courses. So that's why most of the data of this topic will be just a refreshment of your knowledge, and probably you need to just remember that for the sake of the other material of the course. Then we'll talk about revealing the epigenome. So the DNA actually is encoded with DNA or genes that provide instructions controlling the activities of all cells. So genes influence our behavior, determine our physical appearance, such as skin, hair, eye color, and as well affect our susceptibility to genetic disease condition. So bacteria actually is a single, if we go through the human, the first subtopic review of the cell structure. So bacteria actually is a single organism. Human have like 75 trillion cells of around 200 different types. And cells, as you understand, can vary in size from tiny bacterial cells into very long neurons that we have. All cell types contain DNA except only one cell type, which are the red blood cells. The genetic material affect behavior, physical appearance, susceptibility to diseases. What about cell structure? The major three players in cell structure is the plasma membrane, the cytoplasm, and the organelles. And for plasma membrane, it's a double-layered structure of lipids and proteins that surround the outer surface of the cell, while the cytoplasm is the inner content of the cells between the nucleus and the plasma membrane, while the organelles are structured in the cell that each of them do its specific function. For prokaryotic cells, like bacteria, they have no nucleus, that's why it's called prokaryotic cells, and no organelles except ribosomes. And you can see from the figure, it's a very simple, have a cell wall, plasma membrane. Sometimes it have flagella for movement. Most of the bacteria will have a capsule for protection, and the region occupied by the nuclear material or the chromosome, it's not a nucleus, so we call it nucleoid region. And most as well of the organelles, or all of them, are actually ribosomes. So here you can see the DNA, it's an electron micrograph or EM figure, show the shape of the DNA when isolated from the Escherichia coli. And if you already knew before this knowledge that bacteria have actually two types of DNA. They have like a circular DNA, which is called the plasmid. You can see the bacteria have a lot of plasmid, and it has as well a major chromosome DNA. But all of these are not confined within a nucleus. Here you can see a simple difference between prokaryotes and eukaryotes. The cell type is true bacteria. I mean the examples. Here the eukaryote include protists, fungi, plant and animal cells. The size is a matter because you can see that the size actually can range from 100 nanometer to 10 micrometer in prokaryotes, while in eukaryotes it's a bit larger, from 10 micrometer to 100 micrometer. Of course, the structure of the prokaryotes have no nucleus, and the DNA is actually located in the cytoplasm and no organelles except ribosomes. While in eukaryotes, as you all know, that DNA is enclosed into a membrane-bound nuclear envelope, and there is cytoplasm with many organelles. So for eukaryotic cells, which include plant and animal cells, they have double layered membranes, have a nucleus in many organelles like nucleus, mitochondria, endoplasmic reticulum, and Golgi apparatus. The plasma membrane actually highly fluid and dynamic in structure, and they form a closed compartment around cellular protoplasm to define the boundaries of the cell. These plasma membrane have a very important function, which is selective permeability. So it's permeable, but it shows what goes inside and what goes outside. And they act as well as a barrier to maintain the difference in composition between the inside and the outside. And this function is mediated by specific proteins, which are the ion channels and the transporters. The plasma membrane can exchange material between the extracellular environment through the exocytosis, endocytosis, or even the gap junctions. So to summarize its function, the plasma membrane function in protection, communication, selectivity, responding to the environment, and recognition. And here in this figure, you can see all the different organelles and their function within eukaryotic cells. You can see that the DNA is bounded inside the nucleus, bounded by a double membrane, which is called the nuclear envelope, that have pores to facilitate the transfer of proteins from the nucleus to the cytoplasm and vice versa. Okay, so these actually the structure of a eukaryotic cell. Here you can see as well the difference between a plant cell and animal cell. Actually, the major difference as well you can see in the presence of a cell wall in plant cells and the presence of chloroplast in plant cell, which is lacking in the animal cell. This is one of the major difference. And the third major difference is always the plant cell have a large vacuole, large central vacuole, which is lacking in animal cells. In animal cells, you can see numerous small vacuoles. Here as well, this table is very good in summarizing the different structure and function of each organelles of the eukaryotic cell. Okay, and this table as well is for knowledge. As well, this is the rest of the table, which shows the different organelles and their structure and their function. Okay, so it's important to be familiarized by the different organelles, because we will use this expression throughout the course, and you need to understand what is the structure and function of each of these organelles inside our cells. Okay. So in the second subtopic, we'll talk about how they discovered that DNA is the molecule of life. So initially, they got a strain of a special kind of bacteria that causes pneumonia, S. pneumonia, and they call it the S strain. We'll know why. So this S strain, it's called S because it has a smooth surface. Okay. So they injected this S strain bacteria into a mouse. They suddenly found that this mouse died because basically it got the disease, and that's why it become dead. So they've done another experiment. They got another living R type of bacterial strain, which have the rough R because it have a rough surface. It don't have this capsule, which make it smooth. And these living rough cells, when they injected this type or this strain of bacteria, was harmless to the mouse, so it didn't die. So sure, there is a difference because since the only difference between the two setup is the type of bacteria. The S strain led to the death of the mouse, while the R strain didn't lead to the death. So they tried to elaborate more. So they got the S strain and they done something like heat inactivation or heat killed. So they killed this type of strain of bacteria by heat. They overheated the bacteria until it become dead. And then they injected this strain into the mouse. They found the mouse alive. So sure, now they understood now that sure the heat is a major factor that lead to the inactivation of this bacterial strain, and this strain become harmless. But what is interesting now, they got the they got the hold on they got the heat-killed S strain, which already proven to not leading to death, and they mixed it with the living R cells. If you remember, these living R cells doesn't lead death to the mouse. So they mixed both of them and they injected the mouse again. They found that the mouse injected with heat inactivated S cells mixed together with live R cells died. And this was a surprise for the scientists. And they found as well that live S cells could be detected in the blood of the dead mice. So this result is a very clear demonstration that there is a process of transformation occurred, that the living R cells took in the DNA from the dead S cells, leading to its transformation and became virulent and can lead to death. So let's talk about the DNA structure. So the building block of DNA is nucleotides. Each nucleotide is composed of a pentose, a 5-carbon sugar called deoxyribose, plus a phosphate molecule and nitrogenous base, which are A, T, C, and G. Nucleotides are joined together to form a very long strand of DNA, almost 2 meters, and each DNA molecule consists of 2 single strands that join together and twist around each other to form a double helix. The nucleotide in strand are held together by phosphodiester bonds. And we must understand that each strand has a polarity 5' and 3'. The two strands of DNA molecule are held together by a lot of hydrogen bonds formed between complementary bases. So that A pairs with T with 2 hydrogen bonds and G pairs with C with 3 hydrogen bonds. The two strands are anti-parallel because their polarity is reversed. What about gene? Gene actually is a specific sequence of nucleotide that provides cells with instructions of senses of a specific protein or a type of RNA depending on the cell type. So you can see here how the you can see here this is a single strand, and this single strand, each single strand consists of nucleotides. Each nucleotide consists of a 5-carbon sugar linked to a phosphate group and linked to nitrogenous base. And you can see that A pairs with T with 2 hydrogen bonds, while G pairs with C by 3 hydrogen bonds. You can see the direction here starts from 5' 5' end to 3' end, which is reversed on the second strand, which is reversed into a 5. That proves it's anti-parallel. You can see as well here a more simplified structure of a DNA showing the pairing of the nitrogenous base and the double helix structure. You can see as well each DNA have like major grooves, each major one, and the minor groove. And these grooves is important to facilitate the interaction of proteins with DNA. And actually the structure you can see down below, it is the structure of a messenger RNA, which is a single stranded. This one is not double stranded. So let's talk about the chromosome structure. Each cell contains DNA with million bases, 3 million bases. It's around 2 meters if you stretch the DNA inside each cell, 2 meters. So the chromosomes are highly coiled and tightly condensed packaging of DNA and proteins. And it occurs only, the chromosome, you can see it only during DNA replication. Actually, there are 22 autosomes and 1 sex chromosome. The chromatin material, which is inside the nucleus, is consists of a DNA plus DNA binding proteins, which are the histones. It is a state of DNA inside the nucleus when the cell is not dividing, because when the cell is dividing, the DNA appeared in the form of a chromosome. So the chromosomes, actually the nucleic acid, actually it is the nucleic acid molecule, which is the DNA, which is highly condensed, while the chromatin actually forms of a DNA plus histones or some non-histone proteins, and collectively is called the nucleosomes. During DNA replication, the cell divides by mitosis, so that a 2n cell can give a 2n cells as well, 2 daughter cells, each of them is 2n. While for sex cells, they divide by meiosis, or the reduction division, in which the double complement of the nuclear material is reduced into half. And probably you knew by now that the mechanism of division is a semi-conservative replication, in which the daughter cells receive, each daughter cell receive one strand from the parent cells. So replication occurs so that each daughter helix contain one original parental DNA strand and one newly synthesized DNA strand. So here you can see, starting from here, let's have a pointer. Starting from here, you can see a DNA double strand, which actually contains a lot of gene. These DNA, in this figure, we need to understand how the DNA can be transformed into chromosome, which is actually the highly condensed structure of a DNA, which exists only during cell division. So the DNA, which actually around 2 meters in length when stretched, to be packaged, it starts first level, it's wrapped around histones to form the structure of nucleosomes in this structure, which is called beads on a string. Upon further folding and condensation, it leads ultimately to the chromosomes. And the chromosomes actually consist of 2 chromatids. You have 2 arms, P arm and Q arm, and there is a constriction in the middle, which is called the centromere. Normally, the extremes of the chromosomes are called telomeres. You can see only the chromosomal structure, you can see in the figure, when the cell is dividing. Here you can see the structure of the beads of string, which actually these one, nucleosomes, attached together, and these the beads on string, and you can see under the electron microscope. Let's have a more clear description about this important process of condensation and packaging of DNA inside the nucleus. If we start with the DNA that we call naked DNA, which actually contain, scientists have discovered it contains around from 20 to 25,000 genes. It is around 2 meter long, and you need to package these 2 meter long in a nucleus which have around 10 micrometer in diameter. So as we said, it start wrapping around proteins, which is the histones, to form the structure which is called the beads on a string. These nucleosomes, actually it is wrapped DNA around histones. Then after that, a second level of condensation to form, which actually wrapping of the nucleosomes around each other to form the 30 nanometer fiber. And why it's called 30 nanometer fiber? Because the thickness, the DNA now have been shortened and thickened. In a higher level of condensation, these 30 nanometer fiber are rolled around each other to form loops of 30 nanometer fiber. Okay, again with a high order of hold on, higher order of chromatin folding. You can see now it's highly condensed, and this is the shape, and people and scientists now try to understand how these folding occur. And now the chromatin, you can see it inside the nucleus. So this is a brief description of how condensation of the DNA inside, or packaging of the DNA inside the nucleus. You need to understand the difference between the beads on string and the 30 nanometer fiber. Scientists have managed to have an electron micrograph of the different shape of the beads on string and the 30 nanometer fiber. You can see that the thickness of the condensed DNA is more than beads on string. And you need to understand as well that genes, in order to express their product into or got transcribed into messenger RNA, they have to be in the form of a beads on a string, because once they are in the form of a 30 nanometer fiber or more, these genes cannot be transcribed. Okay. So you need to understand again that the nucleosome, actually it is a structure of a histones, which are actually a protein or a DNA binding protein, in which the DNA is wrapped around these histones to form the structure of a nucleosome. So let's talk about now the karyotype analysis, and they use this karyotype analysis to understand the structure and to detect the abnormality in the chromosome. In a karyotype, the dividing cells, because it must be dividing, because we have already have said that the chromosomes is not cannot be noticed or figured or seen only except in a dividing state of the cells. So in a karyotype, these dividing cells are spread onto a glass microscope to release their chromosomes. And then these chromosomes can be stained and aligned to create a basotype, a karyotype based on the overall size of the chromosome, the position of the centromere, and the staining pattern. So you can see that now they can be aligned using a computer software, and actually they are can be stained by computer software as well. This is a normal figure without being stained. It's like another process of staining called the banded stain. So they only stain the chromosome with a stain that gives dark and white bands, as you can see. So you can see here the difference between two karyotypes. A normal karyotype of a male, and how did I know it's a male? Because you can see the XY chromosome here. And this is a karyotype of a HeLa karyotype. So what's a HeLa karyotype? HeLa, actually it is a type of cancer cells from the uterus. So this is a cell that from the uterine cancer, and it's called HeLa. And why it's called HeLa? Because these type of cells have been isolated around in the 50s from a German lady who got her uterine cancer, and she donated these cancer cells for research. So for that, they gave the initials of her name. Her name was Helen something Lavernac or something like this. So they give these type of cells HeLa, HeLa. Okay. So again, in this karyotype, the both karyotype showed the difference between the normal karyotype and the HeLa karyotype, or the uterine cancer karyotype. If you look closely, you'll find that there is 3 copies of chromosome 1, which is normally 2 copies in normal. You can see only 1 copy for chromosome 2, 1 copy for chromosome 3. You can see 3 copies of chromosome 7, 1 copy of chromosome 6. You can see 3 copies of chromosome 10, 3 copies of chromosome 12, and as well, and goes even 4 copies for chromosome 15. Okay, and of course this is a female because it have XX. Okay. So here we can see how they can do a karyotype. They initially, let's have a pointer. They initially collect the blood is collected and got centrifuged to separate the blood cells from the fluid. And now they have the blood cell. Then from the tube, the fluid is discarded, and you need to add a hypotonic solution is mixed with the blood cells to enable the cell to swell and got burst, so that the white blood cells, which actually contain the DNA, will release their DNA into the outside after the destruction of the white blood cells. So you do another centrifugation step to get down all the cell debris. You can see the cell debris all together, and you pour the content over a slide, and then you put a stain to visualize from this drop of cell suspension is poured on the slide, and you can, as I said, you can put a stain to stain these chromosomes. The slide can be viewed over a monitor or a microscope to identify and to align the chromosome together the same size, and to identify the homologous chromosomes. And now the result can be displayed, and you can see from the result these are 46 chromosomes, actually 22 pairs of autosomes, and the sex chromosomes lying each together each other. And this is actually a basic step, the basic steps of doing a karyotyping. And as I said, karyotyping is very useful in identifying any abnormalities in the chromosomes.